All right, welcome back to another explainer. Today, we're totally bypassing the basics and diving straight into the raw mechanical divergence of cell division. We're talking mitosis versus meiosis. It's a process happening inside your body right this very second, but as you'll see, the paths these cells take are just vastly different. So here is our roadmap for today. We'll start with the two paths of cell division. Then we'll hit mitosis, the somatic copier. Third, we'll break down the anatomy of a chromosome. Fourth, we're getting into meiosis, the gamete generator. And finally, we'll look at how all this ties into the human biological clock. Okay, let's jump right into part one, the two paths of cell division. So cell division actually splits into two entirely different operational paths. Think of them as the clone path and the unique path. And this fundamental divide really comes down to somatic cells versus germ cells. Somatic cells, your skin, your organs, your bones, they are built entirely for growth and maintenance. Germ cells, on the other hand, well, they are strictly for reproduction. And the actual biological machinery used to divide each one is completely different. Let's actually look at the somatic side first. Which brings us to section 2, mitosis, the somatic copier. I always like to think of mitosis metaphorically as the reliable, great copier of your body. Its one and only goal is to make a perfect, identical duplicate. This happens in those somatic cells we just talked about through a process called karyokinesis, which is really just the scientific term for the division of the nucleus, and it happens in a very strict 4-step chronological order. The sequence kicks off with prophase. This is where the nuclear membrane breaks apart and the DNA condenses. Centrioles move to opposite poles of the cell, deploying these tiny spindle fibers. Flowing right from there into metaphase, the chromosomes line up perfectly down the cell's equator. Then anaphase dramatically pulls them apart into daughter chromosomes. And finally, telophase comes in and wraps those newly separated chromosomes in fresh nuclear membranes, creating two distinct nuclei. Moving right along to section 3, anatomy of a chromosome. Now, before we watch the rest of the cell split, let's take a quick detour to look at the actual machinery being separated, just so we don't get lost in the jargon. The absolutely crucial structural element here is the centromere. It's defined simply as the exact location where two DNA parts join. You can think of it as the critical anchor point for literally everything that happens during division. And where this anchor point is located actually dictates what kind of chromosome we're looking at during anaphase. It's actually a beautifully organized classification system based on this one feature. If the centromere is dead center, it's a metacentric chromosome. A little off-center, that's submetacentric. If it's just below the tail, it's acrocentric. And if it's exactly at the tail, it's telocentric. Pretty straightforward, right? So to wrap up this whole somatic copying process, telophase is immediately followed by cytokinesis. That's the physical splitting of the cell's cytoplasm. The end result? Two daughter cells holding the exact same number of chromosomes as the original. The copier has done its job, producing a completely perfect biological replica. All right, section 4, meiosis, the gamete generator. So what happens when the goal is reproduction rather than just making an exact replica? Well, we pivot to a much more complex branching pathway. Unlike the single run-through we saw in mitosis, meiosis actually requires two distinct rounds of division. We call them Meiosis I and Meiosis II. And each round has its very own karyokinesis and its own cytokinesis to achieve the final goal. And here is the absolutely crucial takeaway. Mitosis yields two identical clone cells, right? But meiosis, because of this double division process, results in 4 unique haploid gamete cells. Haploid just means they carry half the normal genetic information, which makes perfect sense because they're designed to eventually merge with another gamete. But that brings up a really interesting question. How exactly do they become unique? Well, the real magic happens in Meiosis I, specifically inside Prophase I. Before the chromosomes detach, they actually swap characteristics through a gauntlet of 5 highly specific substages. Ready for this? We've got leptotene, zygotene, pachytene, diplotene, and diakinesis. This sequence is literally the ultimate engine of genetic diversity. It wildly shuffles the deck of traits before the cell divides. After diakinesis, cytokinesis I occurs, and that completes the first half of the whole process. From there, we move straight into Meiosis II, and honestly, this is where things get a bit more familiar. The second round of nuclear division, karyokinesis II, runs right through Prophase II, Metaphase II, Anaphase II, and Telophase II. It's strikingly similar to mitosis. The major difference here is that Prophase II doesn't have those 5 complex substages we just talked about. There's no more exchanging of characteristics. The genetic deck has already been shuffled. Following this, cytokinesis II kicks in, finally giving us those 4 totally unique haploid daughter cells. Okay, let's zoom out a bit for our final section, Section 5, the human biological clock. We've spent all this time zoomed way in on microscopic machinery. Let's pull back and look at the actual human lifespan, because there is a massive, fundamental difference in when this whole gamete-generating process actually triggers, and it depends entirely on whether you are male or female. Honestly, it's one of biology's most surprising reveals. Get this. In males, the process of meiosis doesn't even begin until puberty, so typically around 12 to 13 years of age. But the female biological clock operates entirely differently. In females, the process of meiosis actually starts before birth. Yes, before birth. But it doesn't finish. The cells enter that super complex prophase I stage and literally hit pause right during the diplotin phase. They sit there, completely paused, for over a decade. That makes diplotin the longest phase of meiosis in females by far, finally unpausing and completing only at puberty. Just take a second to think about that. The cellular machinery making up future generations is initiated before a mother is even born. It sits dormant on a microscopic level for over a decade. If the diversity engine of female meiosis literally pauses before birth, what does that incredible timeline mean for the broader human life cycle? What does it mean for our genetics and our overall development? It's just a profound biological marvel to consider. Thank you so much for joining me for this explainer, and I'll catch you guys next time.